welcome to another Piper Pod. Today we're going to start in pathophysiology, and we're going to start with the fundamental aspects of pathophysiology, and we're going to look at certain things that change cellular structure. And the reason that's important is in order to understand pathophysiology, so the basis of disease, it's a good place to start is looking at different types of manifestations and different types of disease patterns that occur within every disease. So every disease will have a component of cellular change or cellular involvement. So before we move on to specific types, we'll look at the general, so the thing that affects all diseases, and then we'll move on to things like inflammatory and then move into systems. So we'll start the process off by looking at the pathology of the cell. Now, as most of you are acutely aware, there's a whole bunch of different cells within the body. So the picture that's in front of you represents a normal or standard cell, and it contains organelles within it that are purely placed for demonstration purposes. So no cell actually looks like the picture in front of you. The reason it's there is so we can talk about each of the organelles. Now, a bunch of them you already know, and we've gone through a few of them, especially in anatomy, physiology, and protein synthesis, things like that. So we'll just move around the cell and we'll talk about each particular one. So the nucleus and the nucleolus contains all of our DNA and our chromatids. So remember, a chromatid is DNA strands when it's during the interphase, so essentially during normal cellular function in the cell cycle, before it's been replicated and turned into chromosomes. So that's where all the DNA is housed. Attached to that we have the rough endoplasmic reticulum and its associated ribosomes. This is the housing area of internal transport of messenger RNA. Mitochondria, you're all aware, powerhouse of the cell. Full details of mitochondria, check the lessons on metabolism processes. Now, other things that we have talked about before is the smooth endoplasmic reticulum and the Golgi apparatus and its associated Golgi vesicles. Now, these are responsible for movement in the cell of different compounds, proteins, things like that. So the smooth endoplasmic reticulum will move components to different parts of the cell, while the Golgi apparatus will package them up in vesicles to go through endocytosis out of the cell. Now, a few structures that you may not be aware of is your intermediate filaments, your microfilaments, microtubules. Those ones in particular, they're mostly responsible, believe it or not, in just housing the cytoplasm. So they form what's known as the cytoskeleton of the cell. Now, obviously, the cytoplasm is the liquid aspect within the cell, and the plasma membrane is a semi-permeable structure that is used to keep the cell at bay and allow movement of ionized charged. Your centrosomes are responsible for cellular division, so very active during mitosis. And then you move down to your perioxiomes. Now, these are interesting little vesicles that are associated with fat metabolism, so breakdown of fatty acids and the repair of the plasma membrane. Lysomes we've talked about heavily during phagocytosis. They're responsible for containing digestive enzymes to break down particles, or in terms they get used in a process known as apoptosis, which we'll talk about later in this lesson. And the last one, which is not very big in a eukaryotic cell, it's mostly associated in plant cells, is your vacuole. Now, this is just a housing unit of water. Okay, so that's your cell organisms. I just wanted to give you a overview on those because a lot of the times we forget the basic cellular structure before we move on to the pathophysiology. So every cell in the body has the ability to communicate with other cells and communicate with itself. And what I mean by communication, it's a means of sending out chemical signals in order to stimulate a response. So very similar to the way drugs work, the chemical process is produced by the cell and then released into the system to either stimulate itself, neighboring cells, or a completely different target cell. And this is where the term autocrines and paracrines and endocrine actions come into play. So autocrines are chemical signals that will stimulate a response within the same cell. So will get released by the Golgi apparatus to stimulate a different receptor on a cell to have a response. Paracrines are neighboring cells, so they get released in order to have a stimulus across a cell mass nearby. Now this is particularly important because obviously when you think about it structurally wise, the cells create tissue, tissues create organs, organs create systems. So paracrines are a way of making sure the entire tissue is having a similar reaction. And endocrine, well, you obviously know that's massive in the hormone, and we're not going to talk too much about it because there's going to be a whole topic on pathophysiology within the endocrine system. So let's move down into cell injury. So that's a normal happy cell and what they do. Now a cell injury is simply anything that interferes with the biochemical processes. Now cells are responsible for a huge amount of biochemical processes, mostly cellular respiration is the massive one. Protein synthesis is another huge one. So anything that occurs that interferes with those processes is considered a cellular injury. On top of that, anything that changes a cellular structure, now this can be either a malignant growth, be either damage to the cell, so put under certain stresses, making the cell grow an abnormal way, or it can also be as simple as changing ion channels. So something that comes along and changes ion gates within the cell, therefore changing the homeostatic relationship between ionization of that cell, is also an alteration within that structure. Now on top of that, a lot of those top two go hand in hand with triggering some form of mechanical response. Now this can be the release of keratin, it can be the starting of the proliferative response, inflammatory response, histamine response. All these responsive mechanisms are associated with cell injury. And then in the end, there's obviously anything that causes your cell death. And we'll look at each one of those as we go through the different pathology states of a cell. But before we get to that, we'll look at the options of what happens every day within your cells. So we have normal cells, and every day your normal cells are put under stresses and they go through a version of adaptation. Now we'll talk more about adaptation in a little bit, and we'll focus just on cellular injury for now. So when a stimulus or a negative influence comes in and causes cell injury, a cell injury can be what's known as reversible or irreversible. Now a lot of the time, what determines whether or not a cell injury is reversible or irreversible depends firstly on the duration, nature and severity of the injury. So a constant duration of the same stimulus, or a violent stimulus, or a particularly cytotoxic stimulus is obviously going to result in an irreversible injury, whereas minor of those things are more likely going to be a reversible. Now it also depends on the type of cell that we have. So not all cells are created equal. Certain cells are very adaptive, like muscle cells, and other cells are very unadaptive, like your neural cells, brain cells. So the type of cell can take different types of stresses before it is determined to be irreversible or reversible consequences. And on top of that, it also goes into play of what aspect of the cell life cycle that cell is at. So every cell has a ordained life cycle before it needs to be replaced. That's where mitosis comes in. And normal cellular replacement and cellular death is where apoptosis and that sort of thing comes in. So if a cell is almost at the end of its life, then minor stresses is going to cause the inability for adaptation or fixing itself to go back to a normal cell, and will most likely end up in cell death. So that's the difference between reversible and irreversible. Reversible injuries will just fix themselves. You get a little bit of cellular swelling, a little bit of cellular alteration, it will fix itself and it'll go back to a normal homeostatic cell. This happens every time you go for a run in the absence of oxygen. Okay, it builds up acidosis, the cells go under cellular injury because of the acidotic state, but it's easily reversible. The Cori cycle picks up all the waste, the oxygen changes the acid level, you shift a few ions around, and all of a sudden you're back to normal homeostatic state. Irreversible injuries, well, we'll talk about more when we move down into actual cellular death. Now, different types of cellular injuries focus on different aspects of the cells. A lot of them have to do with its function in aerobic respiration, development of ATP, and its normal processes of creating protein synthesis and moving substances around the cell itself. So anything that causes an injury on those usually is associated with higher rates of cellular death. Now, the big cellular injuries, these ones. So hypoxia or anoxia is obviously a big cause of cellular injury. Now, that is obviously low or absence of oxygen within the cell. Now, microbes we could spend a lot of time talking about because they're the basis of a lot of disease processes. Inflammation alters cells and usually results in cellular death. We have our strong immune reactions, and then genetic and metabolic disorders are obviously associated with changing cellular structure, so therefore resulting in cellular injury. So if the recipe for producing proteins is altered or the recipe for cellular respiration is changed, then we have a toxic cell state. Now, speaking of pure toxins, we have different versions. So you have a direct one, so they're usually associated with your heavy metals, mercury, lead, and what they do is they break your sulfur bonds within the cell membrane and then break it down. But you can also have indirect methods, which is just metabolized toxins or metabolized substances that produce a toxin. So metabolism of protein produces uric acid, which is turned into ammonia, which is a toxin. Now, the last one obviously is stress, which your muscles go under, and that generally causes rapid adaptations. Now, your tenocytes are classic for that, and that's where you get your tendinopathies that occur. Now, with most of these, when we look at our hypoxia versus anoxia state, there is a chance of what's known as a post-perfusion or a reperfusion injury, and that's where the body's been working in an anaerobic state for a particular period of time, and that's going to build up a huge amount of free radicals. So this occurs when you have a blood clot, which happens a lot more than people think. So you have a blood clot in one of the small vessels, causes that anoxic state. It builds up free radicals. Now, free radicals is just another way of saying that they're highly reactive molecules, so which usually is associated with stealing oxygen or coming from oxygen. Okay, so your acids and oxidizing substances are what's known as free radicals. Now, normally, if free radicals are released in the body, we have substances that clean it up. Okay, and that's where your antioxidants come into play. But in a pathological state, these free radicals react with the cell and they cause damage to the protein or nutrient synthesis within the cell, and it causes the cell to go through cell death. Okay, and that's classic in our reperfusion type injuries, and that's where we have a blockage and then the blockage is released, but then all the free radicals that have been produced in the absence of oxygen are now delivered to the tissue. Let's move on to cellular adaptation. So this is as a response to one of the cell's injuries, or a stimulus that causes the need for adaptation. So if you remember the picture, you get from a normal cell, you get a stress. And now stress doesn't classically mean the release of cortisol, that sort of thing. It can also mean just repeated stimulus on a particular area of tissues. So when you have that, you'll go through cellular adaptation. So we'll just go through each of these. Now these are your classic ones. The top four, the top three really, because normal's normal, are more of your general pathological states, and the bottom two are more associated with neoplasmic formations. But atrophy is the decreasing in cellular size. So this happens physiologically, predictably, associated with age. It's also associated with inactivity, which is somewhat predictable. Now, I'm not talking about breaking a leg and putting it in a cast. That's more of a pathological state. But just stop training, that sort of thing. Then you're obviously going to have atrophy of certain muscle groups. But when we talk about it pathologically, it's usually a result of lack of nutrients, a lack of good flowing oxygen, or denervation, so demyelination to a certain area, or nerves getting kinked off, so therefore the stimulus can't go through in order to keep using those tissue, and obviously your inactivity, which causes a decrease in the size of the cell. Now when this happens in the brain, that causes quite a pathological state for the person, and this is where your gyres will actually have channels, and the signals aren't as smooth. Now the opposite is your hypertrophy. So this can be a good thing in the case of skeletal muscles and training, and it also can be a detrimental thing when you're talking about your heart. So hypertrophy of the heart as a result of prolonged hypertension, so you have that massive pressure within the afterload, making the left ventricle enlarge. Now you may think enlarged muscles function more, but you need to remember that the left ventricle is a cavity. So as it enlarged, because of the pericardium and the fibrous pericardium, it doesn't grow out. It takes a lot of that space on the inside, and therefore causing a problem. Now, hypertrophy is often associated with hyperplasia, and hyperplasia is just an increase in cellular number. So a lot of pathological states will cause hypertrophy and hyperplasia at the same time. Now, hyperplasia can be normal when you look at the menstrual cycle, the hyperplasia of the endometrium as a result of estrogen. That's a normal state before it goes through shedding. Another classic one that's not normal but quite common is benign prostatic hyperplasia, and that's where the prostate will actually enlarge because of increasing the number of cells, and will cause interference with the bladder. Now, other ones that you all get every now and then is your calluses or your hardened areas of your cells because they've gone under certain stresses, or walking too much, lifting weights, whatever it is. Okay, so that's a classic example of quite benign hyperplasia. I mean benign as in not something to really worry about. Now, metaplasia and dysplasia is more associated with neoplasmic formation. So I'm not going to talk too much about them because the subsequent lessons will be all about neoplasmic formations. But just so you know, metaplasia is where you have a different type of cell, so different type of cell structure growing or placing itself in with normal cells. So it creates a miscommunication between cells and the need for more rapid adaptation. And dysplasia is it's even worse. It's where the cells completely lose their perfect structure, their formation, so it interferes with function, and they generally is associated with rapid hyperplasia of those dysplastic cells, which means you end up getting these rapid benign growths, things like that. That's what dysplasia is. Now, the bottom point when we talk about intracellular accumulation, that's an example of cellular adaptation, but it's as a response to exogenic or endogenic materials or metabolism. So what we mean by that is if a substance is brought into the body en masse. So smokers, people that work in coal mines is a classic example of a exogenic material that causes cellular adaptation. And that's where the macrophages within the lungs eat the coal, eat the carbon-based gases that are being brought in, and then end up dying and tarring the lungs. So the tissues of the lungs get a black appearance, and that's intracellular accumulation. But other examples can be your lipid stenosis or atherosclerosis, things like that, where your tissues will need to adapt to a constant influx of lipids or plaques or protein, in the case of protein amyloidosis. It's essentially a way of saying that if your cells are constantly bombarded by an external stimulus, usually a normal nutrient, then the cell will go through rapid adaptation to change the tissue structure in order to better function in that particular environment. Now, obviously, those things grow into different pathological states at time. Whilst it's a normal result of adaptation, not all adaptations are a good thing. Okay, so that's intracellular accumulation. Right, moving from adaptation, so in the event the cell cannot fix itself or cannot adapt to an environment, then it will go through what's known as cellular death. Now, there's two different types of cell death, and we'll go through each of them. We have necrosis, and then we have apoptosis. So, first of all, necrosis is a negative stimulus or a pathological or undesired state results in unplanned cellular death. Now, the different types of necrosis that you can get are listed in front. So your coagulative necrosis is your most common, absolutely most common state, and this is a result of hypoxia, so lack of blood flow, and also the build-up of toxins or free radicals will cause coagulation necrosis, and that is your classic necrosis. Now, when we move down to liquefactive necrosis, that is a result of mostly phagocytosis, okay, where the cells will actually engulf themselves and die, but it's where the cellular membrane is broken down. Okay, so when we have a very strong acidic or alkaline state, it will actually break down the phospholipid membrane of a cell, and you'll get liquefaction of the membrane. Now, this is unfortunately quite classic in brain tissue. We'll see, you won't see. So the absence of seeing any cell is, you'll know that it's liquefaction of necrosis. Now, a special type of necrosis, caseous, is caused by mostly tuberculosis, and this is where the cellular structures will die, but they'll hang around. So it ends up looking like cottage cheese, essentially, when you look at it under a microscope. Now, there's certain fungi that will do this as well, but this is where they infiltrate the area and they use you as a biodegradable source, eat you up, and therefore the cells will kill themselves. Now, in cases like acute pancreatitis, we'll actually go through an exzymatic fat necrosis, and that is where a huge amount of lipid-based material will engulf the cell, stopping all function, causing the internal structures to die, but all you'll see is a big fatty deposits. So acute pancreatitis and also your fatty liver deposits are your classic places where you'll see that side of the house. Okay, so they're your different types of necrosis. And this is, like I said, a unprogrammed or an unnatural cell death, as opposed to apoptosis, which is generally cellular death on a programmed state. So things that are normal apoptosis is when we start looking at embryonic development, hormonal functions like the menstrual cycle, we'll go through stages of apoptosis. So with embryonic development, a classic one is you'll actually start developing a hand and you'll have web fingers. Now, as you grow inside the womb, your hand will go through apoptosis and those web aspect of the hand cells will die and they'll change. So other classic examples are bones that replace themselves, and that's just normal shearing down of unwanted tissue. We'll go through it. There's cells that are programmed to go through cytosis. So as I mentioned in the production of B and T cells in anatomy physiology, any one of those that aren't quite perfect, so they end up seeing self as opposed to non-self as the threat, then they get programmed for normal apoptosis. Because if they don't, then that's the formation of a lot of autoimmune diseases. And then there's the cell life. So cells die. So red blood cells, 120 days, classic, they go through your apoptosis. Now there are some pathological states that cause apoptosis to happen in cells that is not physiologically. Now this is a result of cells, mostly during mitosis, that will have misformed or damaged DNA. So during the G2 aspect, where the cell is getting checked before mitosis, if the replication isn't perfect, or if they start producing faulty proteins, then they'll die. Okay, but they'll die in a state of apoptosis as opposed to necrosis. Now we'll see the difference that means for the actual body. Now there are special infections like HIV and viral hepatitis which causes apoptosis as opposed to necrosis as well. So if they're both resulting in a cell death, there has to be a fundamental difference as opposed to one being programmed and one being not. And this slide here represents what that difference is. Now, normally when you're dealing with necrosis, it's rare that you're talking about a single cell, okay? Because you have an exogenic injury which causes a group of cells or in the tissue to go through the same problem. Now, when they go into this cell injury and they can't recover, then you start getting swelling of the organisms and the membranes start to bleb, so just to form out and not function as they should. From there, as the injury gets worse, you get a build-up of internal free radicals, the pH of it changes, the nucleus either degrades, we'll go through different changes of the nucleus later, and the organelles cease to function. As a result of that, the cell goes in a state of distress. So it starts to release these inflammatory properties. So your prostaglandins, your cytokinase, all that sort of stuff gets released from the cell, which causes a pathological inflammatory response. From there, you have a pathological state. So inflammation is now kicked in, and all the things that come with inflammation occur as a result of necrosis. Now, when we look at apoptosis, either physiologically or pathologically sound, it's a little bit different. This is where the cell will actually condense itself, and it will reduce all the sizes of its organelles. It will start breaking down the actual organelles for function, so they'll start recycling itself, almost digesting itself. From there, it will turn itself into a bunch of smaller cellular fragments, and those cellular fragments, or apoptotic bodies, will go through phagocytosis, and as a result there is no inflammation. Okay, there's phagocytosis, but in this case the phagocytes will not release any cytokinase in order to produce an immune or a inflammatory response. So it happens without the second, third effects with the rest of the body. So that's the difference between your apoptosis versus necrosis. Necrosis is very violent and messy and causes the body to react, apoptosis is smooth and programmed and almost a secret to the rest of the body that it's occurring as well. Now, the last thing we'll look at, just for purely interest state, is when the cellular injury is irreversible, so it's going to end up as cellular death. The nucleus can go through different changes. Okay, so the first change that it can do is the nuclear fading. Okay, so that's your karyolysis. We all know that this sort of thing does happen during prophase of mitosis, where the nucleic wall or the nucleus wall will break down in order to release the chromatids. But in this stage, it's now dissolving as a result of a pathological problem, and that is releasing all the chromatids and DNA into the rest of the cytoplasm. Next one is nuclear shrinking, so the nucleus will compact down onto itself. Okay, so pycnosis is where the nucleus will actually shrink onto itself and become a basophilic mass. Okay, so same as you'll see within your basophil cells. Okay, it becomes like that. Now from there, as it condenses down, what can happen and what generally happens with that is the nucleus becomes karyorhexis, which means the nucleus itself breaks apart and becomes fragments. Now, it's not overtly important that just this happens, because the end result is the nucleus will no longer function as a normal nucleus, and the end result will be cellular death. But for those people that want to go into more study and more interest of different pathological states, it's just an introduction to the terms and the nuclear changes that can occur within the cell. Okay, so that ends the lesson. Like I said, it's just an introductory one. We're not talking about any particular disease at this stage. We're just talking about the general formation and changes of the cells and therefore the tissue. So then we move on to the specific injuries. We can have a better appreciation for what's happening at the cellular level. Until next time, take care.